Hiya! Right, this is our final video for Unit 1, we are nearly there, and it is finishing off respiration with a process called fermentation. The picture that you can see displayed there, a lot more complicated than we are actually going to cover.
Okay, so in this final video for Unit 1, we are going to learn about the process of fermentations and the conditions needed for it to occur. So by the end of this video, you should be able to name the molecule that transfers energy throughout the cell, but hopefully you can already do that. You should be able to describe the main stages of fermentation in animal cells and in yeast or plant cells. You should be able to describe the conditions required for fermentation to occur and state how many ATP are produced via the fermentation pathway / just the general process of fermentation is what that means.
Okay, so fermentation is another type of respiration. So we got aerobic respiration is a type of respiration, fermentation is a type of respiration, and it takes place only in the cytoplasm of the cell, and it occurs when oxygen is absent. Okay, so in our last video where we looked at aerobic respiration, oxygen had to be there in order to do aerobic respiration. In this case, it has to be a lack of oxygen. Okay, so oxygen needs to be absent. Now, it depends on what type of living thing you are, what the products of fermentation are going to be, what are the things that come out at the end of fermentation. In aerobic respiration, everything produces carbon dioxide, water, and ATP. In fermentation, it does depend on what type of living thing you are, if you're an animal, a plant, or yeast.
Okay, so this is starting with fermentation in animal cells. So very specifically in animal cells, starts really similarly to aerobic respiration. It's glucose broken down into 2-pyruvate, and then we've got that kind of swooping arrow again saying that we've got our 2 ATP there. And then what happens if there is no oxygen available, so it is fermentation, the 2-pyruvate then become something called lactate, which is the thing that builds up on your muscles if you go for a run or you've just been at PE or the gym or whatever and you feel your legs are getting really, really heavy. That is because you had a big build-up of lactate. So 2-pyruvate become lactate, but what is really important to note about this is that is reversible, and then you'll see an arrow a bit like that. You'll have one arrow pointing up and one arrow pointing back down. That shows us that a process is reversible, and what that means is that basically lactate can be converted back into 2-pyruvate, and that means if oxygen becomes available, aerobic respiration can then go and happen. If this wasn't possible, it means every time, or that first time you went a run and you got a lactate build up in your legs, if that wasn't reversible, you would have that pain for the rest of your life, and that would be utterly useless having that. So fermentation in animals, glucose to 2-pyruvate with 2 ATP being made, and 2-pyruvate then becomes into lactate, which is reversible.
Now, if we compare that against aerobic respiration makes many ATP. It makes 2 ATP in stage 1, and then in the second stage, it makes many ATP. In this one, you only get that 2 ATP produced. That's all you get. So what we're looking at is a rubbish version of respiration. I like to think of it like if you're in a building and the power goes out, the emergency lighting kicks in. Now, the emergency lighting is rubbish. It is dull. It's not very well lit, but it's kicked in as an emergency measure, and it allows you to get out of the building without falling down the stairs. Okay, that's about all it lets you do. Similarly, in cells, is that fermentation is just keeping the cell with a tiny supply of ATP in the hope that the oxygen comes back before it dies. Okay, so it's keeping you running on very basic functions, very simple functions, for long enough, maybe a few seconds, until the oxygen comes back, and then the cell can get back to business as usual.
What you just said, yeah.
As you just said. Okay, now this is exactly the same as the previous slide, but again, it's in words. Now you need to be able to describe either, so you need to be able to look at a diagram and go, aha, substance X is lactate, or substance Y is pyruvate, or you might just get a question saying, describe fermentation in animal cells, and this is what you should write. So step 1, in the cytoplasm, glucose is broken down into 2 pyruvate molecules. Enough energy is released here to make 2 molecules of ATP. Now that should seem very familiar. If you have watched our last video on aerobic respiration, that sentence is, I'm going to tell you, I copied and pasted. I didn't retype this out. Okay, so that should seem familiar. Step 2, there's a lack of oxygen. So we've got our key criteria for conditions here. There's a lack of oxygen, and that means pyruvate is converted to lactate. Easy.
Okay, so this is our other type of fermentation. This is when it occurs in plants and yeast. So it happens one way in animals, it happens a different way in plants and yeast. Starts off the exact same way as we've already seen. So glucose is broken down to 2-pyruvate and 2 ATP is being made off of that. But what then happens when there is no oxygen available is that 2-pyruvate gets broken down to ethanol, which is basically the chemical name for alcohol, and CO2, which is carbon dioxide. Again, knowing that that CO2 is the same as carbon dioxide. And crucially, this is not reversible. So whereas in animals it becomes lactate, which is reversible when oxygen becomes present, this is not reversible. Because if you think about it in humans, the lactate, if that lactate isn't reversible, you have heavy legs for life. Plants ain't going anywhere. Plants don't need it. If plants end up with ethanol and CO2 and they're staying in the same place, it's fine. They can have this build-up. That's actually how a lot of our foods and drinks are made. This is the process in how most alcoholic drinks are made. So beer, you hear beer is made through the fermentation process. This is what has happened. Yeast is brewed, it's made sure that there's no oxygen, so as it will ferment, and that produces ethanol, which is that alcohol. Similarly with making bread, yeast is fermented, there will be ethanol produced, but there's also CO2. That's what makes your bread rise. So if you watch Bake Off, you'll see that. They put their bread in the proving drawer, their bread is starting to rise. That is because it is not getting oxygen, the yeast is fermenting, it's forming CO2 gas bubbles, their bread is going to rise. So that's how a lot of our food and drinks are actually made through this process of fermentation of yeast or in plants.
Okay, now this slide is just exactly as before. So it's the same as the immediate slide before, but it's in word format. And again, you need to be able to transfer each skill across. So looking at step 1, in the cytoplasm, I'm getting bored of saying this, in the cytoplasm, glucose is broken down into 2 pyruvate molecules. Enough energy is released here to make 2 molecules of ATP. That should seem really familiar. I just said it about 3 minutes ago for animal cell fermentation, and I said it before for the aerobic respiration. So this step 1, this is the thing I'm trying to get into your heads here, the step 1 is the same for all forms of respiration. So all forms of respiration do step 1 in the cytoplasm, glucose, 2 pyruvate, 2 ATP. Step 2, however, for plant and yeast cell fermentation, there's a lack of oxygen, pyruvate is converted to carbon dioxide and ethanol. Easy enough.
Okay, so again, the role of enzymes here, similarly to in the aerobic respiration, fermentation is controlled by enzymes, which means it can be affected by temperature and pH. Again, if you forgot this, go back and look at the video on that and you will see how temperature and pH affects it. So really low temperatures means there's going to be a really low enzyme activity rate. Really high temperatures means the enzymes are going to become denatured. Similarly with pH, really low pH, really high pH can cause the enzymes to be denatured. If enzymes are becoming denatured at all, the rate of fermentation is non-existent. The enzymes are not working, so fermentation ain't happening. If it's just at a really low temperature or a lower or high pH that's slightly off the optimum, the activity of those enzymes, so the rate of fermentation, is just going to be lower. And again, this can quite often be shown in a graph, like your standard temperature graph where it's going up a slope and then a quick decline, and similarly with your pH graphs that you have seen. But again, absolutely crucial, you know what happens to enzymes and how they play their role in respiration.
Okay, now you might need to know this. I think in terms of chemical fermentation, probably not, but we use fermentation. It is useful for us. We use it to produce beer. We take yeast, we give it glucose, we shut off its oxygen supply, and then it will produce alcohol, so ethanol and carbon dioxide. We also, it's what allows beer to be alcoholic. Now, high alcohol drinks, like 40% and above, they have to go through something called distillation, because yeast starts to die at about, it depends on the type of yeast, but about 5 or 6% alcohol, yeast starts to die, which is why most beer is about 4% or so. It depends on the species of yeast that they use. And this is because alcohol is toxic. Ethanol is toxic. If you're a small single-celled organism, you are going to die being swamped by it. This is why we use ethanol as a cleaner. It kills bacteria. This is why over the COVID crisis, we've been rubbing it all over our hands, because it is killing things. In smaller, lighter concentrations, yes, we can use it in a recreational context for beer, but the fact is it's still a poison. And so when this yeast builds up with the when the alcohol builds up in the fermentation, eventually the yeast will die. It will run out of ATP and it'll get poisoned by its own alcohol.
And comparison of the types of respiration. So we've discussed aerobic respiration, we've discussed fermentation when it's in animals, and we've discussed fermentation when it is in plant and yeast cells. So here's a wee comparison table. It's really useful to know the difference. Sometimes you get a question where it'll say, tick which stage of respiration it happens in. Is it in aerobic? Is it in fermentation here, or is it in all of them? So in comparison, for first of all, aerobic respiration, it produces many ATP. Again, you don't need to know the number, you just need to say many or lots, just the idea that there's more than two. The raw materials required, so the things required for it are glucose and oxygen. The things that are produced are carbon dioxide and water, and it occurs firstly in the cytoplasm and then in the mitochondria.
Fermentation in animal cells only produces 2 ATP, so it is rubbish. The raw material is glucose, so it only needs glucose, it's fuel, and the product is lactate, so that's stuff that can build up in muscles, and it only happens in the cytoplasm.
And finally, fermentation in plant and yeast cells, again, only produces 2 ATP. Its raw material is glucose, and its products are carbon dioxide and ethanol, and this occurs in the cytoplasm.
And that is it for fermentation. So the molecule that transfers energy throughout the cell will be ATP. You do the next one.
Okay, to describe the main stages of fermentation in animal cells and plant and yeast cells, well, in animal cells, it's the idea that glucose goes to 2-pyruvate whilst 2 ATP is produced, and it is then broken down into lactate, which is reversible, whereas in plant cells,
in plant cells, glucose converted to 2-pyruvate, 2 ATP are produced, and ethanol and carbon dioxide are produced.
I do the whole of aerobic respiration. It's only
I know,
fair.
I'm smiling the fact that I gave you the big one.
Yeah. The conditions required for fermentation to occur is the idea that there is no oxygen present, and also the idea that there is a optimum kind of ideal range for the temperature and the pH for the enzymes.
Yep. And the number of ATP that are produced in fermentation pathways is the same for both types. It's 2 ATP is that's what's produced. So not enough to stay alive forever, but enough to temporarily stay alive until the oxygen comes back. And that is it for Unit 1. Okay.
I wish I had like a little party popper.
I know, some sort of banner running across the screen. Can't be bothered. So that is Unit 1 of the whole of
Nat 5
biology. So that's a third of the course that we
have
done.
There are still 2 more units. There will eventually be videos for them.
They'll be posted probably after October, the October holiday. We'll probably spend a lot of our holidays doing that. Yay.
It'll be multicellular organisms next, and then life on Earth finally. That is cell biology complete. That is the tricky one because it's all the microscopic stuff that you kind of have to learn to visualize and understand without actually seeing these things. And the next topic is more about the kind of human biology side of things, except from one random rogue bit about plants in it.
Yeah.
But predominantly human biology. So hopefully this has helped. This helped a lot. Go back, watch these again, get really bored of our voices.
Get
to the point
onto the
you've memorized it.
paper website, and for goodness sake, try doing those. Look through your PLPs. We'll probably be posting, well, I'll be posting stuff on Google Classroom for my individual classes. But yes, if you're not a member of my classes, then sorry, that's not going to be accessible to you. Why are you watching this video if you're not my student? That's just weird.
Other people would want to watch it because it's helpful.
Really? Oh my goodness. Okay.
I don't know. It's going
to
be
well done to you for surviving that. But yes, is that lots of revision required. It is a difficult area. The main thing is, it might seem like it makes sense now. You will forget this. Trust me, you are going to forget all of this stuff. Revise.
Everyone does.
Go back over it on a regular basis. In fact, right now it would be a good time to go and look at cell structures and remind yourself what cell structures are, what their functions are, that sort of thing.
And if you think these helped, please say, because there is no point in us sitting wasting all our time making these if you think these are utterly useless.
Although don't tell me that, because that will be offensive. Yeah. I won't like it. Anyway, good luck. We will see you for Unit 2 in a month. Bye.